Okay, continuing our next section of notes, let's talk for a second about non-nuclear inheritance. So if you're watching this lecture right now, you actually have already done the assignment on Google Classroom that sort of led you into an understanding. It was a bit of a video study. You watched a couple of videos, you answered some questions, led you to an understanding of what in the world I mean when I say non-nuclear inheritance. So this should be a pretty quick run through, and then I'm going to finish out the video by talking about ways in which the environment impacts our phenotype. So the last section of notes, essentially. So non-nuclear inheritance. This, to be very, very clear, this is DNA that you inherit through something other than the nucleus of mom's egg and dad's sperm. And so when we talk about non-nuclear inheritance, that really is DNA inherited by way of the mitochondria. And then your video study didn't really talk about it, but pretty much everything that we talk related to mitochondrial DNA applies for chloroplast DNA as well. So mitochondria and chloroplast, they have their own little bits of DNA, and those can be inherited. They are, if you check out this picture up here, this is a eukaryotic cell. We've pulled out a mitochondria, and inside that mitochondria are these small circular components of DNA. So essentially, this is DNA. This is genetic information that you are inheriting from a parent that isn't in the nucleus of their cells. So non-nuclear inheritance. Now, mitochondrial DNA, which is abbreviated mtDNA often, sometimes I've seen it just abbreviated mDNA, they drop the T, but mitochondrial DNA has a couple of characteristics, a couple things that we need to talk about. Remember, what I'm addressing with mitochondrial DNA in concept applies to chloroplasts as well. So we are animal cells, and we don't have chloroplasts, so we're going to focus on the mitochondrial DNA, but the same kind of general ideas is going to apply to chloroplasts. So where does it come from? Mitochondrial DNA is inherited from your mom during the process of fertilization. So let's look at this picture for a minute. So imagine you have an egg cell. So here's mom's contribution. Here's the egg cell. Here's the nucleus with mom's DNA inside. And here's the mitochondria in mom's egg cell. Here's dad's sperm cell. And so the sperm has all of the nucleus and the DNA compacted right here in the head. And then there is this, it's called a midpiece. And we talked about the cell as a unique cell type back in our cells unit, but just as a reminder, the middle here is called the midpiece. That's where dad's mitochondria lives. And then we have the sperm's tail. And we talked about there's lots of mitochondria here to power the motion of the tail. Well, when this sperm fertilizes this egg, the only part of the sperm that really gets in is the head. And so the midpiece and the tail get ejected. And so that means that when we look at the fertilized zygote, the only part of dad's sperm that entered the egg cell and contributes to the growing offspring is the DNA. His mitochondria did not come in. So all of your mitochondria come from mom's. And so you can see that in this colorful diagram.
Mom, the glitch is happening in the iPad.
Okay, just wait outside and I'll come fix it. Okay, sorry. So the yellow mitochondria up here and the blue mitochondria here, all that you see and get passed down is mom's yellow mitochondria. So essentially, dad's sperm doesn't really give anything other than DNA. It doesn't give, dad's sperm doesn't give a ton more than DNA. Some mitochondria might get in. In fact, a study a couple of years ago, 2018, I believe, said that, you know, if mom contributes 500 mitochondria, dad contributes 5. And so the primary inheritance for mitochondrial DNA is maternal or mom, which means we can track something called maternal lineage. So we can track based on your mom, we can track the flow of DNA from mom's mom, if that makes sense. And that leads back to something called mitochondrial Eve. So ultimately tracking back to a recent common ancestor, sort of the first female human that gave rise to all other humans. And so that video was in part of what you looked at in the video study prior to this lecture. Now, when it comes to non-nuclear inheritance, mitochondrial DNA has a couple of connections that I'll point out. So all of this came from your video study. The first thing is that if that DNA is mutated, it can cause diseases. Just like our nuclear DNA mutated causes diseases, there are mitochondrial diseases as well. And so mutated mitochondrial DNA can lead to, you can kind of look at this diagram, can lead to a host of conditions or kind of issues that could arise. Mitochondrial DNA damage has been found at higher levels in people with Alzheimer's and Huntington's. That's a category of conditions called neurodegenerative diseases. And so I'm not saying that mutated or damaged mitochondrial DNA causes Alzheimer's and Huntington's and other neurodegenerative conditions, but I am saying that it seems to be found in individuals at a higher rate in individuals with Alzheimer's and Huntington's. So there could be a bit of a connection. It might not be a causation. It might not cause it directly, but it could be connected to some of these neurodegenerative conditions. And the third thing here is that mutations and damage to mitochondrial DNA has been pretty significantly connected to accelerated aging. And so there's some interesting studies out there about accelerated aging and looking at mitochondrial DNA. Okay, so be aware, wrapping up this section of our notes, be aware that you do have a little bit of genetic information in every single one of your cells that doesn't live in the nucleus of that cell. It lives in your mitochondria. And how did it get there is a throwback to something that we've already talked about, the endosymbiotic theory. Remember, we think that mitochondria, present day, were probably a long time ago free living bacterial cells, which is why they have their own DNA. And that is sort of an understanding that we talked about in our cells unit. You saw revisited in your video study yesterday from Lynn Margulies and her kind of ideas on the endosymbiotic theory. So you have a little bit of DNA in all of your cells that is outside of the nucleus, and therefore we talk about non-nuclear inheritance, DNA that you have inherited outside of your nuclear DNA. So DNA specifically in your mitochondria. Moving into the last section of our notes, it's time to talk about something that can be pretty cool, the environment's impact on a phenotype. So I alluded to this in class when we talked about hydrangeas, and we'll see a couple of examples here, but this is essentially addressing something called phenotypic plasticity. So phenotypic plasticity is what happens when individuals with the same genotype express a different phenotype. And usually that's because the environment is having an impact on the expression of the phenotype. And that might be two different individuals in a population. It might be the same individual as the environment changes. And so that's a great allusion to kind of what we're talking about here. This is let me go back. This is the Arctic fox, and the Arctic fox actually changes its coat color as the seasons change in its environment. And so we'll talk about how the environment really can impact what we look like and our expression of traits. As it relates to the environment, let me sort of define the environment can be an external thing like temperature or chemicals, the exposure to light. Environment can also be a cellular environment. So where your cells exist. So what kinds of hormones and chemicals are your cells being subjected to? could also be something that we talk about here with environmental impact. This particular example on the screen is the result of something called seasonal melanin. And so melanin is producing the pigment in the fox's coat. And as the temperature changes in the environment, the melanin production either increases when it's warm and gives this sort of reddish brown color, or decreases when it's cold and produces this white color. Now, what that does for the fox is allows it to exist in its environment, sort of blending in a bit better, right? Which is going to help camouflage, deterring any predators that might be after it, but it's also going to help it camouflage to become a better predator and catch prey. And so seasonal melanin here is shifting the color of the fox's coat based on the temperature of the environment. Here are a couple of other examples, and really we talk about this very much as a concept by looking at some examples. And so I would, in your section of notes, it's not just knowing what it is, but it's knowing a couple of examples so that you might be able to write about it well on the AP test. So here are those hydrangea flowers that we talked about in class. Hydrangea flowers will bloom different colors based on the pH of the soil that they are grown in. So this sort of shows you a full spectrum of colors. They can be kind of a blue to a purple to a pink color. And so here's the pH range. If you grow these hydrangea flowers in acidic soil, so around a pH of 5.5 or lower, they're going to be a bluish color. If you grow them in a slightly less acidic, so 5.5 up to neutral, which is 7, they're going to be more of a purple. And if you grow them right around neutral, 7, they're going to be kind of a pink color. And so adjusting full color patterns can easily occur by increasing or decreasing the pH of your soil with chemical additives. Put things in your soil to adjust the pH. If you're really good, you can even hone in on a spectrum, so pink, and look at this range of pink that you can get. So the bottom line here is the genotype is the same for all of these flowers, but the environment is impacting how those genes are being expressed. Here's another example, this Himalayan rabbits. And so there's an initial kind of interesting study done where they took Himalayan rabbits and they raised them in 2 different temperatures. They raised them in a really cold environment, and then they raised them in a really hot, not really hot, but a warmer environment. And what they found is that those that were raised in colder temperatures developed dark pigments on sort of their extremities, so their ears, their nose, and their feet. And those that were raised in warmer temperatures did not. And so think about your own body. These are the parts of your body that get cold pretty quick, right? So heat loss is very rapid in your extremities because the majority of your blood is circulating here, trying to keep your core warm when you are cold and when you are in a cold environment. And so the reason that these extremities developed a black pigment is really to soak up and retain as much heat as possible in that cold environment. It was not quite an issue over here because this Himalayan rabbit was not in quite that cold of an environment, didn't have to worry about losing heat through extremities, and so didn't need that black pigment. Now there's also, so here is that dark pigment is helpful to preserve heat in the cold climates. This is an additional sort of experiment done on a Himalayan rabbit. So they took one of these Himalayan rabbits that was raised in the cold environment, one that they knew could produce that black pigment as seen on the nose and ears and feet and tail, and they shaved a little spot on the back of that rabbit that originally had white fur. And they put an ice pack on the back of this rabbit over a prolonged period of time. And lo and behold, when they removed the ice pack and the fur had grown back, it grew back in this dark pigment. So they were able to again show how the environment right in that area where hair was growing, where new cells were dividing and hair was being produced, that hair grew back darker because of the presence of the cold environment or the ice pack. The last example that we're going to look at here is something that I think is pretty cool. This is, again, the environmental impact on a phenotype. The environmental impact here is temperature. And the phenotype is determining whether the organism is going to develop as male or female. So this is called sex determination in sea turtles. And really, this happens in reptiles in general. I just found an interesting connection to sea turtles that we're going to look at. So essentially, sea turtles do not depend on the presence of X and Y chromosomes to determine sex like we do, but instead, sex is determined by the temperature that the eggs are incubated at as they are growing inside of the eggs. And so females will develop if the eggs are incubated in warmer temperatures, and males will develop if the eggs are incubated in cooler temperatures. So think about in nature, where are sea turtle eggs incubated? In the sand, right? You've probably at some point seen a female sea turtle climb her way out of the ocean, kind of dig a hole on the beach and lay hundreds of eggs into a little bit of a nest. And those get covered up a little bit with sand. And so the eggs continue to mature and incubate in the sand. So if that sand is warm, then the majority of those eggs will develop into females. If the sand is cooler, the majority of those eggs will develop into males. And even temperature changes within the nest, within the clutch, which is what it's called. All those eggs together is called a clutch of eggs. Depending on your position within the clutch, you might have eggs in one place in the clutch that receive warmth and develop into females, and then eggs that are in a cooler spot in the clutch that develop into males. So again, the environment, the temperature in the environment is impacting the expression of a trait. That trait is sex determination. Now, I found an interesting connection here. We're going to watch this video. It's just a couple minutes long about temperature, present day temperatures, and this whole development of male female in reptiles. So you have to read it. You can't just listen. So check it out by reading the screen. Okay, so a connection to, our climate change, and this is not, it doesn't matter kind of what your personal stance on things like global climate change are. This is a really great example of how the environment has warmed up in some capacity. It has warmed up, and now there are way more female turtles than there are males, which one day could become a pretty significant problem for continuing generation after generation of sea turtles. So using these examples, using the hydrangeas, think about these are all examples of environmental impact on a phenotype. So for the hydrangeas, the environmental impact was the pH of the soil. The phenotype impact was the color of the flower. Then we looked at the Himalayan rabbit. So the environmental impact was the temperature of the environment. The phenotype expressed had to do with dark hair on the ears, feet, nose, and tail. The last example that we just looked at here, the environmental impact again is the temperature, and the phenotype impact is sex determination. So those are really good examples for you to use and kind of an understanding of how it's not just your genes and what you have inherited. It is also the environment in which you are raised in some instances. Kind of an interesting thought to think about. Okay, you are done with this lecture.